And this is what we're going to talk about, the fact that they're terpenes, what a gibberellin looks like, what they do, and a little bit about gene expression, but not much. Okay. Gibberellins were first identified in Japan right before World War II, so nothing was known in here until after the war. And basically what his observation was when this fungus attacked these plants, the plants would just grow really tall. And that's basically what gibberellin does, is make things grow tall. And they actually isolated the fungus and then did the experiment when they grow the fungus, then filter out the fungus, pour that liquid on the plant responds. So that means there's something in the liquid that the fungus is producing. Okay. And named gibberellins. They are part of the terpenes. If you remember, we talked about terpenes as one of the classes of secondary metabolites. Stem elongation, seed germination, you've had that, right? First exam. Gibberellic acid and breakdown of production of alpha amylase. We're going to have it again. It also is involved in reproduction. Let me talk a little bit about the genetics. Where have you seen dwarf peas before? You've all seen that. Everybody, I bet, has seen this before. You've seen it in high school. If you've had genetics, you've seen it. Who did that? Who was the first person to work with dwarf peas? Who? Mendel. And by the way, his data was too perfect, by the way. There's some people that question whether or not his data was really that good. And he didn't fake it. He may have just threw away some outliers. And so let's talk about genes. This is a gene that is not a mutant. And usually when we talk about genes, and then here are the ones that are mutants, and of course they have less endogenous. You know, if we do this versus a little LE, that the big L, you know, that's the normal, the non-mutant. Okay? And we're not going to go into the nomenclature, but if it's a gene, it's bold, and if it's a gene product, then it's usually italicized. And usually has 3 letters. But the fact is that if we look at these plants, if they have these, if they're recessive, and so when it's recessive, that means both alleles are the little L, right? Then the plant doesn't grow, or it grows in the intermediate stage. Mendel was really lucky in what he picked, and probably he did a lot of other things, but it didn't work out, so he stuck with the ones that he could actually work with. There are a large group of compounds, and they go in and out of being biologically active. GA1 was the first one that was found, and the nomenclature goes up. I don't even think I included it in here. Pretty much they're all the same molecule. They have methyl groups added to them, or they have hydroxyl groups added to them. And of course now I'll show you the structures, but now of course with mass spec, if anybody knows how mass spec works, then it's a lot easier now to figure out what the specific structure is of these kind of compounds. It's in young tissue. And in other words, developing things. It is not that clear if it is actually found in other tissue. It can be assayed there, but it is pretty clear that part of, if not all, the GA synthesis it depends on chloroplast or plastids. Let's say not chloroplast, but plastids. So it's not everything is synthesized in the cytoplasm. And a lot of these things have other components of them. So if you squint your eyes and look at these, they all look the same. GA3 is probably the one that is most used because it's very active and it's not hard to make. Okay? Why do people use 2,4-D and or 2,4,5-T? Why? Why not another oxide? They're cheap. Easy to make. Just have to be sure and get rid of the dioxin before you use it. So they're fairly complex molecules. Here's one called Gib-Grow, and you may have heard, I don't know if I put ProGib in here or not, but it's a commercial form of gibberellic acid that you can use for a variety of things, and I've got some examples in here. GA1 is what we find in plants, GA3 is what we spray on plants. Some things that it does that are really interesting: stem elongation by both cell division and by elongation. Bolting, in other words, if you've got a biennial crop, you spray GA on it and it grows up and produces flowers the first year, so that's good if you want to, if you're a seed producer. Alpha amylase, you should know, does break dormancy, probably the same way it stimulates alpha amylase. In cucumbers, it can induce maleness, I think in general in cucurbits. Parthenocarpic fruiting, what's that? Anybody? You've probably seen that. You get fruit set without seed production. And you can spray gibberellic acid on certain things and cause it to set fruit. I don't know, is that used in cucurbits? Anybody know if it will actually set fruit, cause fruit set? I don't know. It's used for some crops, I know that. And it also can act to delay senescence. So gibberellin is sort of like we call it a growth hormone. Elongation. In this panel, you see on the left, you see a dwarf plant, and then the second one over is a dwarf plant that's been sprayed with gibberellic acid. And you see it's almost normal size, and then you have normal sprayed with gibberellic acid. So what you see, and it's easy to see in corn, we have a lot of dwarf mutants. We used to work with viviparous mutants, which were, a lot of those were dwarfs. You want it, if you want to cross that plant, you spray it with GA so it grows up and you can get some pollen and cross it to whatever you want to cross it to. And it works pretty well. It's pretty easy to do. And of course with the Arabidopsis, there's a whole host of mutants that are available. Here we see not just growth, but what we call also bolting. This is a brassica thing. So you see with more GA, you get more growth and flowering. Here's the classic one. This is what Mendel worked with. On the left, it's a mutant. It's a dwarf pea. And on the right, it's sprayed with gibberellic acid. And so you get quote, normal growth. Easy experiment to do. A lot easier to do with Arabidopsis, of course. It is also important to that compounds are used that stop gibberellin. So why would you want a plant to stop growth? What kind of plant would you want to be really bushy and not grow really tall? Ornamentals, right? So you want a small bushy plant, got a lot of flowers, looks pretty. It can also be used for changing yield of things. Aaron talked to you about barley. These are two you may have heard of, Arest and Bonsai. Here's Encimidol, and from left to right, and this experiment actually has to do with light intensity also, but we're just going to look at the fact that you get a more compact plant. You get an anti-GA effect with this compound. And then we have Bonsai. I think I have an example. Yeah, same thing basically. More Bonsai, the shorter, more compact the plant. Anybody know what a Japanese bonsai is? Anybody know what those are? You get a really old tree that's about this tall, and it's got bark on it, and it looks like it can be incredibly old. They actually go in and pull off the young leaves and go with the gibberellic acid. So it still grows like a tree, but it's literally like that. real bonsai, not have been chemically. You can that is an incredibly ancient and very hard thing to do. To actually pick the right leaves at the right stage, so you end up with literally an old tree that's like this tall or even shorter than that. You've seen pictures of them, and you'll see people out on the side of the road selling them. Those are all been chemically induced. There's not been, no one has spent years picking the leaves off the source of gibberellic acid off and letting the other leaves grow. Yes, sir. I'm sorry, I can't hear you. Yes. Yep, you do have to prune them all the time. I don't know exactly how. I mean, I know that you prune them all the time, but I don't know if I've ever seen a real bonsai plant. I mean, I may have, but I really wouldn't know. Commercial, bolting, so you can do seed production. Did I already said that? Here's a classic picture in plant physiology. One of the first hormones that we started messing with was gibberellic acid. And this is, oh goodness, I can't remember his name. Oh man. Anyway, this was made in Beltsville, Maryland at the USDA facility there. And this is tobacco bolting. So you can probably tell which ones were sprayed with GA and which ones weren't. Quite a difference there. Oh, what is that guy's name? Can't remember, I'm sorry. Malton and barley fruit increase, increased sugarcane yield. Thompson seedless grapes can be made this way. You can tell which one has been sprayed with gibberellic acid, and what they do for grape production is they spray the gibberellic acid. Well, I think I have it up there. Spray gibberellic acid, and causes basically the stems to elongate, so they have more space to grow. Then they spray them again to get better fruit set. So the first spray will actually help thin the fruit set, so you get better grapes. Can't do this with wine grapes, unfortunately. Not as easily anyway. Thompson seedless is a particular kind of grape. So anyway, and this again is a classic picture that I saw when I was an undergraduate taking plant phys. That's why I like to put them in. When I taught lab, we did a bioassay for gibberellic acid. You take known amounts of gibberellic acid, put them on shoots, and they grow different. So you see what we're developing there? We're developing a standard curve, right? So I can actually draw the axes on here. So what you do in lab is you do that, and then you take some tissue and you grind it up and you put it on another set of these shoots, and you see how much they grow. And then you just go over, right? Everybody know what I'm talking about, don't you? You just go and see where it falls on the standard curve, and that tells you how much gibberellic acid you've got in your unknown plant. Works really well. It doesn't work as well as spending $100 in an assay using the antibody prep. But it's like I said, it's good enough for state work. We've done a lot of work in the past with different companies that think they're producing some kind of growth regulator. Again, if you don't purify it, you can use it. So anybody heard of Actinovate by any chance? It's a bacterial product this guy just found, and we did a little work on it, and guess what? It's an auxin. At least if you put it on internodes, it elongates them, which is what auxin does. And if you add an antioxidant, it doesn't work. So whatever he's producing has auxin-like properties. And there are a few other that we sort of looked at. And you can buy all these things. You can buy algae concentrates and all sorts of stuff. There was, I don't know if I told you last time, Friday was 420, which is the big marijuana day apparently, and I was watching TV, and I guess Berkeley's always had this huge crowd on their commons, and they decided not to do it this year, so they sprayed some kind of plant product that's made from dead fish or something, so it stinks so bad nobody would come on to the quad. But I guess if you're all doped up, I don't think you'd even know it, but I don't know. Here's a neat little picture. This is an artificial color picture. What this is a picture of is gene expression. And what Dr. Kiowa does a lot is he adds a fluorescent protein behind the promoter. And so if that gene is produced, it lights up. Okay? So you're not looking at the gene, you're looking at the promoter. And so this is an artificially colored picture, and you can see the red being the highest concentration where it's produced in the youngest tissue. Okay? Neat little picture. Showing you that it's produced in that young tissue, and then of course it's diffusing out a bit. Here's a classic picture again, and what you see, the point of this, is that gibberellic acid induces something fairly neat, cell division. So you think if you've got a developing fruit, even way when it's really, really small, is when the cell divisions occur. And so by the time, again, the only thing I know about is corn, by the time a corn plant's probably maybe even less than a foot tall, all the ears are already there. And all the tassel initials are already there. And almost the rest of the growth is by cell elongation, cell expansion. Due to turgor pressure and softening up the cell walls. Yes, sir.
When wheat grows, like clear wheat, it just grows and doesn't have any stem elongation or anything, and then goes into the reproductive mode and it starts making nodes and starts elongating. Is that like what you...
I would think, I don't know particularly, but I would think. There's a lot of, when we finish up talking about Dr. Borlaug's work but you know, I mean, can you spray something on it to increase that? Do you know what you spray on it right now? That's basically what gibberella does, is it induces the formation, the cell division, which induces formation of organs. So I'm not sure it's auxin and gibberellic acid.
Okay, so when it's like in its dormant winter mode, it's got all those nodes that just all stacked up, and then when it...
That could be auxin too.
Reproductive mode,
It doesn't both. That could be either. It could be probably gibberellic and auxin, gibberellic acid and auxin. I would bet. No, I don't know specifically. Well, you just talked about the fact that there is a signal that acts with a receptor, and you've seen this before on your first exam. I didn't know this was in the book until a couple of days ago. These are all new lectures. It takes me about 6 to 8 hours to do a single lecture. So, and some of these are not from the book, but you saw this on exam 1, you know, and on the left you have corn seeds where they tried to take the embryo out, but they left a little bit of the embryo behind. So you get a little bit, you put it on a media containing starch, and you stain the starch with iodine. And then on the right-hand side, they've added gibberellic acid. And so the alpha amylase is diffused out and eaten the starch, and you get nice little circles. And again, this is great to do. And I should have set this up for class. It's a great little experiment to do in lab. You can add different gibberellic acid concentrations and come back and measure the circle and draw a standard curve. Works really well. Doesn't cost $100 an assay either. Now, here is something that I wanted to put in because it shows you the fact that you have this cascade mechanism, okay? This signal transduction cascade. So you have calcium increasing, which changes the pH can change. That CAM is calmodulin. So the point is that you get a series of things happening, and then you get the alpha amylase produced. Remember from week 2 or 3 or whatever, gibberellic acid, when we talked about gene regulation. So again, you have all these things that are happening in sequence before anything else is going to happen. And then you actually then have a breakdown later on, but it takes a while to get that gibberellic acid. Whoops. And you saw this as something we went over before. On the left panel, we're looking at with or without GA enzyme, and on the right, we're looking at the messenger RNA production. So you see, without gibberellic acid, you don't get messenger RNA production, and you're not going to get the protein being produced either. Now, if you grind up a dry seed, you can actually pull out some alpha amylase messenger RNA, a little bit, but not much. Okay? Okay, and you saw this the last time. Here's one from the book, just to show you again. Gibberellic acid diffuses into the what? The albumin, cool. Where it produces alpha amylase and also a protease to produce beta amylase. We talked about alpha amylase. And alpha amylase does what? Breaks starch down. That starch, either as glucose or maybe even the maltose, then goes back into the embryo and supports growth. Cycle complete. Okay. One of the best and first studied hormone systems in plants. Again, great for an undergraduate lab. And you look where the starch has been broken down, you measure how big that is, and you get a really good idea of how much gibberellic acid's in whatever tissue you're trying to assay. Now, here's something that you may not have seen before. This is actually it's called electrophoresis, and basically it's a gel, sort of like agar, and you can run proteins through it, and you can separate out proteins. And the one you see here is the second panel where it has gibberellic acid, and guess what? You get this protein that is alpha amylase produced. You get a bunch of it produced. And you don't see it in the other. You see it a little bit in the control. You never ever stop gene expression 100% of the time. So even if you pull out the embryo, you're still going to get a little alpha amylase produced. Very few things are off or on switches. G protein may be the only one we've talked about. It's either off or on. And this is something we talked about the first of the semester. Now you can put it all together. We have this gibberellic acid binding, which ends up going in and acting with the DELA genes, which we'll talk about in a moment. Starts then the production of messenger RNAs, which end up being transferred to the endoplasmic reticulum, where the alpha amylase is synthesized, which is then processed in the Golgi, which then moves to the cell membrane. The Golgi blebs off, it moves to the cell membrane and dumps the alpha amylase outside the albumin cells into the starch in the endosperm. Okay? Remember this? Now, let's talk a little bit about the Green Revolution and Norman Borlaug. Did any of you meet Dr. Borlaug by chance before he passed away? Passed away, what, a couple of years ago, maybe? Maybe not even 2 years ago. Real nice guy. The Green Revolution was a lot of different things. It was using fertilizer, and it was effective use of water, and it was improving irrigation. One thing that I wanted to talk about here, though, is the fact that they, as countries, or as CIMMYT and IRRI, which is the Rice Research Institute in the Philippines, started using fertilizer and improved management. The Green Revolution was a whole series of things over a long period of time. But anyway, these especially these wheat would grow up and fall over, called lodging. And so what they did was they actually, Dr. Borlaug, basically this was his work, was he put in genes that were basically semi-dwarf genes into wheat, and I think corn too. So now, instead of a taller plant with fertilizer, you get a shorter plant. So that does two things. Keeps the plants from falling over, and it redistributes the carbohydrate from the stem into the actual wheat grains. So you do two things. You keep it from falling over, and you now have a greater increase in yield because it's shorter. Does that make sense? You're not putting all your carbohydrate into growing that stem and growing up. So it was these kind of, and they're known now, and actually they've just been identified, I think in 1999 they actually identified what the genes were that were in the original varieties that he used. So he started this stuff back in 1952. I want to say when the Nobel Prize in 1970, anybody know? Quite a while. And he won the Peace Prize, of course. Okay? Now, okay, we're out of time. Let me just finish up the slide next time. So, any questions about anything? Send me an email, and I'll post stuff. The exam will be on metabolism and photosynthesis. Yes.